So this is the pan-genome discussion. I've been wanting to do this for a while. I am no expert on pan-genomes, which is part of the reason I wanted to do this discussion. So, you know, it's been lots of years now, we've been talking about pan-genomes, but there's some different views of what this means. And then there's the question of, well, it's interesting, but what's it good for? And then how can the data portals present this in such a way that it's helpful? So these are the questions I want to discuss today. So I start off by defining or coming up with this definitions, multiple definitions of what a pangenome is. And I'm going to show a few really quick examples. They're just links of some that I'm aware of. And then we've got a few people to talk about their work, and then we'll have a discussion. So here are the lightning talks, and let's see if I can still remember how to talk about a pangenome viewer. Alan Cleary is going to talk about his context viewer, and Marcella is going to talk about the pangenome browsers at Grameen. So this is going to be a very interactive discussion. So first off, I'm curious to get people's opinion on what exactly a pangenome is. So it seems like it can be genome or gene focused, can be based on a reference or be an all by all. Can be looking for large or small structural variation across the genome. Is it only within a species? Can it be within a clade, across plants, across animals? And then sort of what conceptually is it? Is it a graph? Is it a set of alignments? Is it a set of syntenic relationships? And then where do you draw the line? When is it a pangenome? When is it just variation data? And is, you know, for example, genotype data from a SNP chip a form of pangenome? So eager to hear what people think about this.
Does this also include alternate assemblies of the same genome by different assemblers?
Yeah, it's a good question. Any opinions?
I haven't really dealt with pangenome data, but I would think it's more of a gene sets from the genome assembly from like various cultivars or various lines, or so that reference genome can have a certain set of genes, but then like other genomes from other variants will have more genes, less genes. So pangenome would be like the complete gene sets. We can't really say complete, but then more like bigger gene sets that can be in a subset that includes like the genes only in a subset of species.
Yeah, like we use older versions just to improve, just to compare annotation, but we wouldn't consider that a pangenome.
Right. Yeah, I wouldn't think so either.
This is Pankaj here. I think how the pangenome has been described by the microbiomes and other communities is a complete picture of a genome of all the structural variations and of all the possible paths on a chromosome by chromosome basis. So for example, rice pangenome, original rice reference was about 430 megabases. But by the time they started putting together a pangenome based on a lot of other genomes from the same genus that was a species, rather at a species level for Rhiza sativa, for example, it turned out to be double the size, so around 650. It's all the possible variations of a of a genome that you start constructing. So it's kind of a graph, I would say, but and you would not have one single serial molecule kind of things. You will have multiple paths overlapping and some things that you would see. So it can come up with something like what you have in hops and things like that, like some kind of a heterogeneity within the genome. So you start having a portion of the chromosome showing heterogeneity. So that's where I think the pangenome is going in direction. About what Suk you were saying and what Grameen was trying to do at that early level is a pan-gene atlas. Well, what is the cumulative set of all the genes present in the genus or in a species level? That is a pan-gene atlas. So those two are different things, I guess. So, but I've seen that many of the pan-genome papers have basically used the synteny and the gene trees to draw the pan-genome graph in that way.
I would say this is Stephen. The answer to your question, what is a pangenome, is yes. That is, to each of your bullets, you can find examples of something that someone has called a pangenome, which points out maybe that our language hasn't matured enough yet, that it could be genome-based with a graph concept, or it could be a set of, a collection of gene sets, could be within a species, or it could be within a broader clade, etc.
I kind of like the term pan-gene atlas.
Yeah.
Okay.
So this is Doreen. So I think that we should think about this much the same way as we think about the genome. So I actually think genetic maps were always a representation of the genome, way before we had reference sequences, and it was our representation of a genome, the markers, and then we had really we had less quality reference sequence based genomes or gene spaces for a while. That was our representation of our genome, and then we had really good reference assemblies, and we have really improved reference assemblies for multiple things. So from my perspective, I think the definition of the pangenome becomes context dependent, because in this case here, we're focusing on a pangenome perhaps for a species or closely related species, but there's also the context when you think about pangenomes, which is a little out of this, of when you think of a plant microbe interaction. When you think about a biological system, and this is closer to some of the microbiome context where you're looking at the gene space that's required for the mechanistic model that you're looking at. So I think all the things that we're discussing are all examples of it. It comes down to what are contexts, and we're defining that gene space as being in what tools or data sets that we have to get to our pangenome space.
No, that makes a lot of sense. It kind of fits in with Stephen's yes. And I think with that, we should move on so we can get through all of the material, but that was helpful for me. Okay, so after this call, I'm going to make the slide set available at the AgBioData.org website. So I am collecting examples of portals, and if you know of any, put them in the chat, and I'll add them to the slides before they go onto the website for sharing. So here are a few. There's cowpea, Brachypodium, wheat has done some really good work with pangenomes. Unfortunately, Dave Edwards couldn't be here, but he sent some information that I've included in the slides. He's, I guess, part of the pan wheat genome. Pan pig, I did manage to find one plant, two actually, pig and goat. But there's obviously a lot of important organisms that are missing that I'm pretty sure have pangenomes. And then here I'm collecting some examples of visualizations that you can get to online or have publications. So if you're aware of one, again, put it in the chat or email me, Effie, or Jacqueline, and I'll get it into the slides. Okay, as we go through these lightning talks and further discussion, remember the key questions are, what is it useful for? Who is it useful for? How can we make the data accessible? And what role do data portals play? So we will start with LOI to talk about his Pan-genome viewer, and then he will correct my pronunciation of his name, I hope, when he comes on. Okay, I'm gonna stop screen sharing and then turn it over. Okay.
Okay, I go then. Okay, can everybody see my screen?
Yes.
Yeah, good. Okay, so hello everyone. Today I'm going to talk about pan-genome visualization, of course, how to think it, and I will introduce a bit Panache, which is a prototype I've made for pan-genome visualization. Okay, so first of all, a little bit about me. I'm a first-year PhD student. Well, actually, I've just celebrated my first-year anniversary of the PhD, yeah. And I'm based in Montpellier, working with the Institute of Research and Development. And my PhD subject is the development of a tool for the visualization of plant pan-genomes. So I'm working on plants and not bacterial pan-genomes, but they have their specific problematics. And through my first year of PhD, I faced, I faced different challenges. The first one is indeed how the visualization can scale to big genomes. You have talked a bit about this before the conf started, but indeed, with the few tools that already exist, there are many that are for bacterial pangenomes, and they do not scale well with plant pangenomes that are way bigger. I couldn't use them directly. Some of them have many problems or are not maintained anymore, which makes it quite difficult to use. Another thing is that there are multiple representations of pangenomes that are available, and this is quite linked to the fact that there are indeed multiple definitions of pangenomes. So you've talked about the gene atlas and the structural pangenome, let's say, and those definitions come with their own problematic too. And sometimes it can be represented as, for instance, a presence-absence matrix. If we talk about genes, for instance, like which gene is present or not in which genome. But we could also represent it as a graph with sequences linked together. This is, yeah, a big question, but most of all, something I want to talk about is that it is impossible for us to see everything at once, despite what some people want to do. And I will explain a bit this. So basically, pangenomes are complex data. There is a lot to see, there is a lot to do, and not everybody wants to do the same thing with a pangenome, which makes the whole thing quite difficult. An example with the graph pangenomes is the hairball effect, where there are too many branches, and this leads to a complete unreadable visualization. But also, the thing is that our mind, our brain cannot focus on everything at once. For instance, here, there is a little typo, and it is easier to see this typo because I've highlighted it with the visualization. All the data, all the text is presented, but you cannot see this at the first glimpse if it's not highlighted. With this, I want to point out that the insight can be gained with visualization, but also data processing. We have to focus on little parts of the data beforehand. We cannot just take all the data and show to everyone. There is work to do to process and to attract the eye on some specific parts. And this led me to think that a good viz tool should have both summarization of information and exploration. By summarization, I mean access to inner properties. For instance, with the Pangenomes, most people are interested in what is the core genome or the dispensable genome, meaning what are the parts that are present in every genome that are part of the Pangenome, and the dispensable genome, meaning the parts that are present in only some of the genomes. So basically what is stable or not. And the exploration is having the possibility to manipulate and to go through different representation of the same data, because different representations means different insights. And with this in mind, I worked on a prototype for a visualization tool for Pangenomes that I called Panache, for Pangenome Analyzer with chromosomal exploration. And I'm going to show the prototype I've made. So here are the links, but you can see this here too if you wish to access it. It's available for everyone. You can just copy paste the URL and you'll find this and you can try it directly. So basically, it's quite similar to a genome browser, but here it's a Pangenome browser because I've added multiple genomes at once. It's based on the principle of a presence-absence matrix, meaning that each genome is cut into different blocks, and those blocks can be present or absent. For this, I've put fake data, so those blocks in here can be either genes or just sequence. It has no importance right now, but it's kind of a proof of concept, you know. And I also added the possibility to highlight which are the parts that are present everywhere at the core genome in orange, or that are present just in some parts, the blue, and to change the threshold of those parts. In this way, we can see directly both in the miniature of the chromosome here, which is orange, or which is blue core or dispensable genome. I've added different things, but the thing is that with such a representation, like kind of a browser, we can add multiple layers of information, such as a classical browser do already. Here is one for the core genome, here is one for the pangenome position, and here is one for the structural variations. So for instance, what I wanted to add is information of about where are the other repeated blocks. So let's say this block here, it's present in the genome 1, genome 5, and genome 6. And we can see that it can be found in some other genomes, some other chromosomes, sorry, the chromosome 0 in strong quantity and the 3. Yeah, that's it. You can play with it a bit, but that's really the proof of concept here. Yeah, this is a prototype, so of course there are missing elements, but I would like to highlight a few interesting of them. So what we would like to do with this tool is to allow the exploration through different zoom scales. Here it was the block scale, kind of, intermediary scale. So we're already looking at a pan chromosome, but we would like to be able to go deeper at the nucleotide level, seeing SNPs of interest, for instance, but also to have a broader vision and to see more how are the structural variations linked together. Another thing is that we would like to add more representations, because as I said, more representations give more insights. And really interesting point that I wanted to highlight too is that when I talked with different plant biologists, they wanted to be able to link the Pangenomes work with what they have previously done, their previous analysis, because they do not want to lose the work, which led me to think that a good thing would be to be able to change the kind of reference systems. I mean, like switching from a pan-genome system to a referenced one, being able to switch the reference with the visualization so that previous analysis could be used again. Because yeah, not many people know what pan-genomes are. Even less people are at ease working with them. So we do not want to lose them in the process. Most importantly, the data. Something that can be quite difficult is that there is no standard format for the genome files. But as you know, visualization and data are directly linked. And one question I have is that maybe we could use multiple data for the different representations of a pan-genomes, because using a whole big enormous file for all the different possibilities of representations seems a bit extreme. And I think maybe we could divide data into different formats to access different information. Yeah, that's all I have to present. I think yeah, a bit too much for the time. But if you want to discuss it further, feel free to contact me. I'd be more than happy to answer your question. And thank you very much.
Thank you. That was actually, got one really quick question. Are you planning to make the code available once you get past prototype stage?
Yeah, and we want to put the code available on GitHub. The prototype is already available in there. Okay. Even if it's not quite up to date yet. But yeah, we want to make it accessible. That's the point.
Great. Yeah, it looks wonderful. Okay, next person is Alan Cleary. You want to take it away, Alan?
Yeah, give me just one sec to pull up my screen here. All right, that should be it.
There's a hole in the middle.
Oh yeah, that's Zoom demanding attention. Okay, present. Okay, how's that?
That's good.
Great. So I'm going to be talking about a tool that I work on as part of the Legum information system. It's called the Genome Context Viewer, or GCV as we refer to it. And I guess before we dive in here, I think Eloa made a really good point about it not being realistic to try and you know, look at the entire content of a pangenome all at once. You're just not really going to get much out of that. And kind of in that spirit, the GCV comes from a completely, it comes at the problem of representing and visualizing a pangenome from a completely different direction. That is, what happens when the data that you want to compare is actually distributed across multiple data providers. Do you leave it up to the user to kind of aggregate the data themselves and then load it up into the viewer offline? Or do you give them some kind of recourse to pull in all the data dynamically and you know, kind of explore it in that dynamic sort of way? And so that's what the GCV is trying to do. We actually developed it as a comparative genomics tool. So it's not explicitly for pangenomics, but it can be used for pangenomics. So that's the context we're going to be talking about it in today. Right, so what you're seeing right here is the context viewer. And there's two main visualizations going on. The one on top is the Macro-Synteny viewer, and the one on bottom is the Micro-Synteny viewer. And the Micro-Synteny viewer is actually what drives the entire show. So we're going to kind of break this down, starting with the Micro-Synteny viewer, specifically that track on the very top. So let's dive in. All right, so the way you load up a genome context is via the Micro-Synteny view. Specifically, you have to provide it with a query track. And the way you define a query track is actually with just one gene. This one in the middle here, we refer to it as the query gene. And you tell the viewer how many neighbors on either side you want your track to be composed of. So with the query gene and the number of neighbors, you end up with a slice of a chromosome that looks like this. So these triangles on a string, it's really just an ordered sequence of genes on a chromosome. The orientation of the triangle represents the strand that the genes occur on. And as you can see, each one of these genes actually has a color associated with it. And so what we're actually doing is coloring these genes based off of their functional annotations. So in this case, the functional annotations are coming from the gene families of the legume federation. And so what that allows us to do is kind of search and compare for similar tracks based off of these homology relationships. But even without searching, we're already starting to see some inter-track homology. So on the right side here, we got a little bit of a copy number going on. Yeah, and just a couple more things about the track. Each one is labeled with its chromosome, as well as the genomic interval that the strand of genes occurs at. And then you can actually get a little bit of insight into the physical distance between the genes based off of the thickness of the lines between the genes. So the thicker the line, the larger the distance. Okay, so that's what a query track is. So this is where the fun begins. The GCV takes your query track and it goes to all the data providers that have implemented the GCV service API with the functional annotation. So in this case, the legume federation gene families. And each one of those providers returns a track that has similar functional annotations. And then using those functional annotations, we provide a local alignment of each of those result tracks with the query track. And so what that does is it kind of highlights the preservation of structure among the tracks, as well as the variation that's occurring. So as you can see, in this case, we have some copy number as well as gene presence absence variation going on. And then we've actually modified these local alignment algorithms to also explicitly draw inversions. So they kind of jump out at you. So in this example here, we're actually looking at soybean, and it's not very compelling from a pangenomics perspective because the inversion is actually occurring on a different chromosome. So it's a duplication that occurred recently. So as far as I understand it, this isn't going to affect recombination or anything like that, but it's illustrative of the types of structures that the GCV can unveil with its searches. Last thing to note on this slide, in the bottom right corner, we have a dot plot. So if those line thicknesses between the genes really isn't telling you the full picture of the positions, the physical positions of these genes, you can actually pull up a pairwise dot plot between any of the result tracks in your query track and get a better idea of what's going on. So in this case, we have the dot plot for the very bottom track here. And the inversion that is occurring is it's pretty apparent. Okay, so the second viewer within the context viewer that we talked about at the beginning there is the Macro-Synteny viewer. So this is kind of similar to the Micro-Synteny view in that it's dynamically loading blocks that are similar to the query chromosome. That is the chromosome that your query track occurs on. So in this case, we're looking at glycine max chromosome 15, and we have some other chromosome 15s popping up, as well as the chromosome 8, where that duplicated block had occurred. And the nice thing about this is it actually puts your Micro-Synteny view into context. So on the right side, you see this kind of semi-opaque bar highlighting part of the view, and that actually corresponds to the Micro-Synteny view that we were looking at. So when you put it all together, you kind of get this picture of where these microsyntenic structures are coming from, or rather, are they just microsyntenic structures? Or are they part of a larger syntenic structure? And what are their physical locations on their corresponding chromosomes? And the interesting about this too is that we can see that there's kind of this break on chromosome 15. We might be curious why that is. You can actually grab this semi-opaque highlighting bar in the Macro-Synteny viewer and drag it wherever you want to highlight a new portion. And that'll load up the corresponding Micro-Synteny view and allow you to inquire into what's going on. Okay, so glycine max, that was just kind of a demonstrative example. Let's actually look at a couple pangenomic examples. So here we have fail, and these functional annotations are actually based off Panther gene families from the Phylogenes project. And as you can see, there's some extensive copy number variation going on, as well as some presence-absence variation. But generally, there's pretty well-preserved structure at a Macro-Synteny scale. Another example we have here is cowpea. And so, as you can see, the viewer can handle much larger tracks than that small glycine example we were looking at. But what's occurring here is a large inversion, and in this case, it is not occurring in a duplicated sequence, so it actually is affecting recombination. And as I mentioned at the beginning, the GCV is actually a comparative viewer, not necessarily just a pangenomics viewer. And so at the bottom here, we've actually pulled in Adzuki bean as well, which is an outgroup of cowpea. And we're basically using it to infer the likely ancestral state of this region. And basically, we're doing that to help avoid orientation bias. Which is a nice feature if you're worried to like oversampling or something like that. And last but not least is something that I can't talk with too much detail about, but it's basically a comment on what you choose to use as functional annotations. So a few of the people on the LIS project have actually been working on they're calling pan-gene sets. And so instead of just your traditional gene families, these are actually functional annotations based off of clustering of genes from the same chromosome within a specific species. And so in that glycine example we were looking at, that recent whole genome duplication was kind of obfuscating the view as far as recombination is concerned. And so with this gene, this pan-gene set approach, you wouldn't have that issue. So basically what we're seeing here is just two cowpea Micro-Synteny views that have the different gene families. So in the first one, the traditional approach, you just think you have some copy number variation going on, whereas in the second, it's coming across as gene presence-absence variation. Okay, so I kind of just plowed through those slides. There's a whole lot more to the GCV than what I've mentioned here. For example, you can actually have multiple query tracks. You're not limited to one. And then another one, which Elwal actually touched on, is that the GCV is looking at things at a very specific resolution, in this case, the gene view. And so it's useful to be able to interact with other tools that are looking at things at a different resolution, for example, a nucleotide resolution. And so GSV is actually very interactive. For example, you can interact in real time with other tools such as GA-Browse via what are called broadcast channels. And this functionality is implemented in the Legume Information System. So if you're curious about that, feel free to inquire or check it out for yourself. And then, yeah, one final parting shot here. We are interested in loading other functional elements into the genome context sphere besides protein coding genes. So if there's anything in particular that you would like to see, let us know. Thanks.
Great. It just gets better and better every time I see it. Marcella, are you ready?
Yes, I'll share. I want to say that my talk is going to be a little bit of an overview, more from the biological, the users, because I'm not a software developer, but in the group we have, right now we have Doreen, we have Andrew and Sharon, if you guys have more technical questions. So basically I'm going to tell you about the work that we have done for 4 different species to get pan-genome browser views using the Gramian ensemble infrastructure. And we have taken advantage of the homology-based type of pan-genomes. We have taken advantage of these, the protein coding genes to build gene family trees. And this is through using the ensemble compara pipelines for building the trees. There's more information over here if you are curious. These are the 4 crop communities that we have worked with for the browsers. It's rice, maize, grapevine, and sorghum. And for each one of those, we have a prototype. And actually, the OG is already public, browser fully public. We have begun, as I mentioned, from the gene space, exploring the gene space. So these are gene-centric browsers, you can say. We already have the collections for reusing the Gramian and ensemble infrastructure. Like I said, the gene trees and the synthetic maps. These are the type of views that you would that you can think of as zooming in or zoom in out of a Google map, just like Alan mentioned, the macro and Micro-Synteny views, where on top you have like this is the entire genome, like each chromosome labeled with a different color for a bunch of them. And you can go into single genes in this example. Let me use the pointer. You would have like the gene-centric view. Oops, sorry. Here with the red line, and then you can have flanking genes to both ends and see how's the conservation to work in a whole clade or in a whole group of genes. This is another. These are other views from the maize example in this case. Same thing on top, you zoom out or you zoom in, and this is to the amino acid level. You can see gene conservation across the members of a single of the pangenome or outgroups, closely related species like sorghum, setaria, and so on. This is a nice tool for us because it allowed us also to identify like potential annotation errors or biologically relevant differences among species. Like in this case, there is what looks like a deletion over here. We also have another type of view that we can that we have with this in the pangenomes is you can zoom all the way in to the amino acid level. And again, you can see conservation to the amino acid level across the entire genome. This already mentioned, and a little more detail. Okay, so I already told you what we had, where we already that is already done. This is something that we're still working on. We are taking advantage of these nice views and tools and more coming new data to improve our annotation pipelines and so on. Oops, and on that and on those terms, like, so I mentioned before that we had gene trees, and we also have whole genome alignments, which are useful to build Synteny maps. So these allow us to identify real differences among different varieties in a pangenome, or potential annotation artifacts. In this case, this is a real one. It's an example of a domestication gene, the shattering one gene in locus in that is absent in the African rice, Glaberrima, but is present, conserved in Oryza sativa and Oryza bati. So that's where the locus is. And in Glaberrima, in the African version, we had an inversion at that same region and a deletion. And in the tree, you can see that there is the YAP2 gene, but it's not the species of the African species is absent there. Now, for the future, things that we are looking for, moving for, are more to at the DNA level to support the differences. We are looking to work with the genome-wide alignments to validate DNA level views. So next steps would be to have to support new views and new ways to search for these, an improved search for this information, taking advantage of all the different types of analyses that come with a pangenome. This is work that we have been doing with support from the NSF and USDA. Sharon is the main software developer for an analysis for the Pangenomes. Andrew had developed the APIs search and the views that I show for the Grameen tree-centric views, homology views that I show. So this is work done at the WER lab, but collaborators from various other projects.
Great. Looks like lots of new stuff going on at Grameen. I'll get your slides and all the links into the presentation so people can go look at that in depth. So at this point, I think probably best to, I'll bring up my slides again. Probably a good time to, well, okay, so first off, when I show this, I'll share these slides. So here are some papers that I found very helpful just in getting basic grounding in Pan-genomes. It's hardly, I'm sure there are other really good ones, but if you're looking for a start. And then I got a lot of papers from Dave Edwards and from Rex Nelson. So I've been adding these here on and on. At this point, I think we should open up to questions for the presenters and more discussion. So there have been some questions in the chat. So if you want to do a little bit more closer questioning.
Hi, this is Sushma, and I have typed a question in chat, and I see that Micro-Synteny can be used perhaps to make the gene nomenclature a little bit more consistent, because we have this challenge that with each new assembly for any species when it comes up, how do we going to keep the gene nomenclature consistent? And as a bench scientist, people remember the gene names and gene standard IDs, and they like to hook on those. So this is the broader question for everyone who is dealing with the pan-genome. So I think that perhaps within the species, what thoughts do you have that some of this data can be used for at least keeping the nomenclature consistent for the set of the genes that are conserved throughout? And then how do we accommodate nomenclature of the genes that are coming out of new assemblies and things like that?
Well, I don't know how much this addresses your question, but one thing we're doing with MAGE-GDB is keeping all of the gene names from the different versions and different assemblies, different accessions, but then grouping them under what we're sort of a pan-gene ID. And I think we haven't got this figured out with the community yet, but I think there may also be an element of adding the classical gene names. Like LG1 will have a version 3 and version 4 gene model, and then it will be associated with gene models across all of the different varieties that have been sequenced. So basically people can use their IDs. We ended up with a plethora of IDs, but people can keep their favorite ID for their favorite genes.
Hi, this is Andrew. I also wanted to just note, I think the Micro-Synteny does a nice job with genes that aren't subject to copy number variation and kind of establishing what the correspondences are across genomes between allelic copies. But certainly there are types of genes, like our genes, for example, where it can be really difficult to decide exactly what the correspondences are between genomes. And I think it's kind of an open question as to what the community really wants in terms of handling pangenes in such cases. It's not even clear that there are necessarily things that would pair up together, if you were to make a cross between these particular lines. So I think it's a complicated question.
I think this is Doreen. I think it's also really complicated because, as I was discussing earlier, we had different representations of granularity of these genes, these genomes, from genetic maps to good reference sequences. But even the reference genomes now are usually just representing one haplotype. And as we are starting to see, some of the other more of the genomes now are representing the 2 haplotypes. We're even having challenges on how we represent a single individual with 2 haplotypes.
I think we lost you, Doreen.
Oh, I'm sorry. I was bringing up that I agree with the Andrew about the challenges with that of the copy number variation and presence absence. But we're also finding, as we're working with these genomes, most of the time my group has been mostly working with inbred lines that are just representing a singular haplotype. And now when we're starting to work with other species, and the granularity of the sequencing coming out, we're actually getting the multiple haplotigs that are coming out from that individual, and we don't even have a good way of representing the 2 haplotypes from a single individual.
Yeah, we run into that as well with, say, a species like alfalfa, which is a obligate outcrossing autotetraploid, and your assembly represents 3 to 4 haplotypes, and if you pick one, you might be picking a haplotype that happens to be missing a gene. So trying to coalesce that information into something representing a sort of within genome pangenome is kind of a challenge. I imagine you have that issue with a grapevine.
Yeah, grapevine is when we've been working with the grapevine community, we recognize how naive we've been.
Yeah. Well, you're lucky it's a diploid.
We've still got about 5 minutes if anyone has a question or something to bring up. Still got some time.
So this is Pankaj here. So very nice presentations by the 3 groups that we saw today. So my question is based on your views and the data that you're looking at, is it possible to create a cumulative or a conservative graph of all the sequences that you've analyzed to say that this is what a pan graph would look like, that has all the possible common and unique genes and genome structural areas in this consensus or a conserved graph that we can think about?
I would say it's a work in progress. And even if we think about right now, even a VCF file is a simple version of a graph.
Mm-hmm.
One of the things that Andrew has been reviewing is, while we are using the infrastructure that we have right now, we know for scalability in the future, we will need to be looking at storing them as graph-based approaches, but we, as Pankaj, as you're aware, we're not at that point. There are other groups looking at that, though.
Yeah, and another one is, how are microbiome guys, the bacterial genome guys, that are handling, because they have hundreds and thousands of those genomes. So is anybody taking a look at what that community is doing?
Rex can jump in here, but he sent several references to pan-genomes in bacteria. I know that they've got, go ahead.
Yes, there's, if you want to see a whole lot of work on pan-genomes, it's in the bacteria. And also we saw a chat about the COVID-19 evidently is also doing a pan-genome. But most of the time it's because they've got such simple genomes, it's sequenced to the gene level, and they just say that sequence is either is either present or absent.
Yeah.
Most of the mutations involved in either virulence or resistance to antibiotics are genes that have come in and gone out. So there's less work or consideration of actual haplotypes rather than just presence absence.
Yep. All right. Thanks. And I do have one more. Is how are people handling the translocations across different chromosomes? Does that count as absent, present, or how do you deal with it?
Pankaj, are you talking about like a single gene translocation or a segmental?
No, a portion of a chromosome just moves to a different chromosome. And so maybe in one or two lines or subspecies or species that moves to a different place compared to all your other
Are you talking?
genomes.
Are you talking for the microbiome people, or are you
No,
talking
no, no.
about for the plant?
No, I'm talking about the microbiome has only one chromosome on that side. That's not a problem. But for plants, for example, the whole section moves in terms of synteny, just moves to a different place. It's not in the syntenic region anymore, but there are conserved gene trees. They're part of this conserved gene trees, but not necessarily part of the synteny. So how do we build, visualize or analyze those breakpoints?
I can speak to that with respect to the pan-gene collections that we've been building.
Mm-hmm.
If a region is moved to another chromosome, it remains syntenic. It's just syntenic as a chunk. And I think that would be true of Alan's context viewer as well. One observation I'll make is I think all of the genome portals will need to be moving toward pan-genes, where instead of a gene page that's anchored on a single assembly, it will be a gene group that you think of as an allelic set across all of your assemblies.
Thanks.
Another thing that I'm curious people's opinions about. So we've seen at least some examples of kind of very gene-centric approaches, and you know there are definitely going to be types of structural variation like presence absence of repeat elements with respect to those genes that are going to influence phenotypes. And I'm curious as to how people are thinking about those sorts of issues.
This is Doreen. We don't have an implementation in place, but we do have the capacity to store the structural variants, right? And so we've been trying to decide if we have them if we query them in context to other functional elements in the genome, right? So we've been thinking about the way a person may want to take a gene set, like the gene tree set that we were discussing, and say, is there any major structural variation associated within a certain distance of the gene locus to support that sort of things. So we do not have any robust implementation in place. We've just been thinking about ways that we could rapidly use searches in context of gene space and functional annotation to actually give you that, right? So if we can have the TEs themselves annotated, we would even be able to query which type class of TE may be associated with that structural variant.
Right, yeah,
So,
we've been thinking about ways of sort of annotating our gene-centric tracks with auxiliary information that wouldn't actually be used to create the alignments, but could be visually representative of those sorts of things.
I don't know if you guys can hear me, but this is Ben Busby. This is something we've thought about a fair amount. And one thing you have to be very careful to do is scope the context of these things. So I think obviously you could actually explicitly represent promoters, enhancers, whatever other elements you want to in a nucleobase graph. So something like VG or AGI. But the problem with that is that then you can't represent all structural changes as a in some sort of reference graph. And the reason is because think about something like trinucleotide repeats. If you represent trinucleotide repeats for a large reference, your graph is eventually going to blow up. So I think when you start thinking about those types of issues, what you want to do are make dynamic reference graphs for fairly small populations. So say you're interested in legumes, maybe you just want to make chickpea. Or even a substrate of chickpea and just make the graphs for that if you're looking at specific genes for, say, drought tolerance, something. So that's something in terms of sort of larger structure of how graphs would be presented to people. That's a consideration to think about. And I think what you could have are some really very approximate reference graphs and some very approximate reference regions, and then really dynamic graphs for specific regions going down to the nucleobase level in specific contexts.
Well, we have reached the end of the hour, and this is a huge topic, but it kind of feels like this has been useful, at least for me. It'd be good if we can, I guess we have contact information for the speakers, and we've got papers, so I think we can probably continue with questions and conversation that way. And this topic will probably come around again before too long. So thank you all.
Thank you, everybody.
Thank you,
Thank
Evan.
Bye.
Excellent presentations.
Yeah.
Thank you. Put it all together.
Thank you.